Good morning, student. Welcome to our new class. Today's our topic is formation of acetyl coenzyme. This is the second step of mechanism of respiration. We studied earlier glycolysis. In glycolysis, we see that from glucose, one molecule of glucose, two molecule of pyruvic acid is formed. Then what is the fate of pyruvic acid? Pyruvic acid is the end product of EMP pathway, or we can say glycolysis. On the basis of availability and non-availability of oxygen, pyruvic acid can be further reducing with different circuits. But here we are seeing that it is in the presence of oxygen, or we can say we are studying aerobic respiration mechanism. Second phase of respiration starts when pyruvic acid formed by glycolysis in matrix. From matrix or from cytoplasm, this pyruvic acid can move to the matrix of mitochondria. One carbon atom of pyruvic acid oxidized into CO2 in matrix of mitochondria. This process is known as oxidative decarboxylation. After this, in the presence of pyruvic dehydrogenase, here the enzyme dehydrogenase. I tell you earlier, whenever from NAD to NADS to NADP to NADPS to FAD to FADS to enzyme comes here dehydrogenase. And whenever we talk about specificity of enzyme, then this enzyme is react on pyruvic acid, so we can say pyruvic acid dehydrogenase. So after in the presence of pyruvic dehydrogenase enzyme, first oxidation take place and then it combines with coenzyme A. Pyruvic acid can combines with coenzyme A in the presence of dehydrogenase, and other 5 factor also come here, just like TPP. Here one marcation is also makes. What is the full form of TPP? Thymine pyrophosphate. Then La lipoic acid and magnesium ion and NAD and dehydrogenase enzyme. NAD convert into NADS2 and NADS2 is the energy binding molecule. Conservation of pyruvic acid into acetyl coenzyme A can be described by this reaction. Pyruvic acid combines with coenzyme A in the presence of TPP, La, magnesium and dehydrogenase enzyme NAD. NAD convert into NADS2 and here acetyl coenzyme A is formed and CO2 is released. We are seeing in the reaction of respiration C6H12O6 plus 6O2 is equal to 6 CO2 plus 6 H2O. So here 2 molecules of CO2, because we know that from glycolysis 2 molecules of pyruvic acid is formed. Then from one molecule of pyruvic acid, one CO2 molecule released, and from another pyruvic acid, another CO2 molecule is released. So 2 molecules of CO2 released here. Then remaining 4 CO2 molecules are releasing in the Krebs cycle. So our next step is Krebs cycle. So here is Krebs cycle or citric acid cycle or TCA cycle or tricarboxylic acid cycle. We can study 2 steps. First glycolysis, second formation of acetyl coenzyme A, and then after Krebs cycle. This cycle is completed in mitochondria and was discovered by British biochemist Sir H.A. Krebs in 1937. In his regards, it is known as Krebs cycle. He was rewarded by a Nobel Prize in 1953. First product of Krebs cycle is citric acid. First product of Krebs cycle is citric acid. So it is called citric acid cycle, or also known as citric acid cycle, because citric acid has molecule of acidic groups. Hence this cycle is also known as tricarboxylic acid cycle. At beginning of Krebs cycle, 2-carbon acetyl coenzyme A transfer both the carbon molecules acetyl group to oxaloacetic acid to form 6-carbon containing citric acid. So it is called citric acid cycle or tricarboxylic cycle. Look at the cycle. First of all, pyruvic acid is formed in glycolysis, and from pyruvic acid acetyl coenzyme A is formed, and here NAD is also converted into NADH2. Okay, and one carbon molecule is also releases. So we can see 1 carbon molecule, 2 carbon molecule and 3 carbon molecule. And what is the reaction? C6H12O6 plus O2 is equal to 6CO2. From 1 molecule of pyruvic acid, 3 carbon molecules are releases, and we know that in the glycolysis 1 glucose molecule is converted into 2 pyruvic acid. So from 1 pyruvic acid to 3 carbon molecules are releases, and from 2 pyruvic acid 6 carbon molecules are releases. Now look at the reaction. First of all, acetyl coenzyme A and oxaloacetic acid OAA means oxaloacetic acid react together and makes citric acid. After citric acid releasing one H2O molecule, remember students, whenever H2O releases or whenever H2O is addition, then the enzyme aconitase we can use here, or enzyme aconitase is in the process. Citric acid convert into in the presence of aconitase enzyme cis-aconitic acid, and you have to write all the reactions. Just like first of all, first reaction is oxaloacetic acid plus acetyl coenzyme A, and then it makes citric acid. Just like second reaction, citric acid is equal to cis-aconitic acid and plus H2O. Okay, so by this sketch you can remember all the reaction, or you can find out, or you can write properly all the reactions by remembering this sketch. Here enzyme synthetase is used. Cis-aconitic acid, addition of H2O in the presence of aconitase enzyme convert into isocitric acid. Isocitric acid release 1 carbon molecule in the presence of decarboxylase enzyme convert into oxalosuccinic acid. Oxalosuccinic acid react with NAD and perform NADH2 and release CO2 and convert into succinyl coenzyme A. Succinyl coenzyme A in the presence of kinase enzyme with the help of GDP. GDP is converted into GTP, and this is the synonym of ATP. So here enzyme kinase is comes. It converts into succinic acid. Succinic acid reacted with FAD convert into FADH2 and makes fumaric acid. And I tell you earlier what, whenever FAD is converting into FADH2, NAD is converting in NADH2, we have used enzyme, which one? Dehydrogenase. Dehydrogenase is the enzyme. And fumaric acid, by addition of H2O, enzyme aconitase convert into malic acid. Malic acid release H2O, and in the presence of dehydrogenase enzyme, NAD convert into NADH2 and it makes oxaloacetic acid. So again oxaloacetic acid combines with acetyl coenzyme and makes citric acid. So these cycles continuously process in the mitochondria, or we can say in the cristae of mitochondria. This is Krebs cycle. Now see the energy budget. We can see energy budget of glycolysis where we obtained 8 ATP. Then from this NADH2 we can obtain 3 ATP, and from this NAD we can also obtain 3 ATP, and I tell you GTP is the synonym of ATP. So here 1 ATP. From FADH2 we can obtain 2 molecules of ATP. This is we can say in the ATS, and from NADH2 we obtain 3 molecules of ATP. Whenever we write down the energy budget, then we write clearly formation of succinyl coenzyme A from oxaloacetic acid, 1 NADH2 is performed. Second step, from succinyl coenzyme A, succinic acid is formed, then we obtain 1 ATP. From succinic acid we obtain fumaric acid, we obtain FADH2, and FADH2 goes into ETS, we obtain 2 ATP, and from malic acid to oxaloacetic acid is formed, then 1 NADH2 is performed, and from 1 NADH2 we obtain 3 ATP. Then total number of ATPs are 3, 3, 6, and 2, 8, 9, and 3, 12. And 3 ATPs are also obtained from isocitric acid to oxalosuccinic acid. 1 NAD is converted into NADH2. Here we also obtain 3 ATP. Then total number of ATPs are 15. Okay. All the counting of 3, 3, 6 to 8, 9, 12 and 15. From 1 pyruvic acid we obtain 15 ATP, and we know in the process of glycolysis 2 pyruvic acid is performed, then 15 into 2 is equal to 30, and from glycolysis we obtain 8 ATP. So total ATP is formed in the oxidative respiration is 38 if the glycerol phosphate shuttle is transported glycolysis NADH2. If malate aspartate shuttle is moving NADH2 from cytoplasm to mitochondria. If malate aspartate shuttle is transported. If glycerol phosphate shuttle transported, then we know that 36 ATP is performed. This is glycolysis, acetyl coenzyme A and Krebs cycle. In next video we will study about electron transport system or ETS. Or importance of this cycle or Krebs cycle is formation of ATP molecules, and they provide energy to different functions. And second, in this cycle many intermediate compounds are formed which are used in other biochemical process. Just like succinyl coenzyme A is initiative molecule of synthesis of chlorophyll, amino acids are formed by alpha ketogluteric acid, pyruvic acid and oxaloacetic acid. Thank you for today.